Thank you for having us again. The purpose of this talk is really to give a brief update on the developments with the COVID Biobank. So as we described previously, the COVID Biobank really integrates several activities across campus to promote research on patients suffering from COVID. Our activities involve active consenting and enrollment of patients into genomic studies. We maintain a liquid biobank, and there's a newly formed tissue biobank as well. We integrate the biobank data and patient data with electronic health record data with the COVID-19 data mart that has been described by George previously. Finally, we are moving forward with genetic and genomic studies. So just to give you kind of a context of what has been done with the biobank in terms of recent timelines, on March 20th, the Department of Pathology was IRB approved to collect residual samples from COVID patients. And on March 26th, we submitted an IRB application to begin remote recruitment of patients, and we formed the structure of the COVID Biobank. On April 1st, we actually enrolled the first COVID patient, and by April 8th, we invited over 500 patients to participate. By April 14th, over 1,000 patients who have been diagnosed at Columbia were invited to participate. And as of April 15th, we started recruitment of inpatients who are able to consent as a systematic activity going forward. So what we have formed was a liquid biorepository that was linked to patient consents, patient self-reported data, which we capture through questionnaires, and you have a linkage to the questionnaire code books that we are using, that we have disseminated widely for the purpose of other studies. We have formed an honest broker system now that links de-identified samples with EHR data, and that honest broker system is maintained by the biomedical informatics resource of the Columbia CTSA. And just to give you an update on the numbers, you can see the sample types that are most prevalent in the biobank. There are some rarer samples as well that I excluded from this table. But as you can see, we have a large number of Buffy codes, urine samples, nasopharyngeal swabs, plasma, and serum. The total number of samples is listed in the second column, and in the third column, you see unique patient counts for each sample. So for example, for Buffy codes, we have nearly 1,700 patients, and we are actively extracting DNA from these samples in order to implement genetic studies. We also, just to give you an idea, there's a large overlap in terms of these samples coming from unique individuals, and out of 1,700, nearly 1,700 patients with DNA available, we looked at how many of patients with DNA also have available urine, nasopharyngeal swabs, plasma, and serum, and you can see that there are fairly large numbers of overlap between these samples, enabling more kind of multi-omic approaches of profiling both host genetic factors, but also biomarkers and other factors, perhaps transcriptomic profiles from these patients, and integrating the data. This is our progress with DNA extractions. We have extracted over 1,680 samples right now. This is data as of April 27th. Most of the samples are actually giving us a high yield in terms of DNA. So we are well positioned to start sequencing these samples using a variety of different methods. And I'll now pass it on to Wendy, who will describe some of the progress on the recruitment and consenting side.
Thanks, Krzysztof. So the coordinators have been busily consenting individuals. As of last night, we had 471 fully consented, another 333 that gave verbal intent to consent, and they're in the process of being signed up. There's a questionnaire that fills in some of the epidemiological data that isn't in the EHR, and we've had a really good response rate to that, 86% that have completed the baseline questionnaire. Next slide, Krzysztof. And as a reminder, those individuals are available to recontact. Just as a reminder for those of you that may want to put in applications, that's the website. It's a simple one-page application to be able to put in requests. And I will say that some of the initial applications came in were relatively broad and non-specific. And the more specific you can be in your application, not to make it lengthy, but so that we can know exactly what you need for your study design, it helps us to be able to fulfill requests. Next slide. As we've been going through this, there's some things that we've realized. And so this is hopefully going to help those of you who have applied or want to apply to get access to your data and your samples better. The first is that some of you have requested clinical data only, and just to put that in the express lane, there's a process called TRACK, which allows you to get data only, so clinical data only that are coming from the EHR, and you can put that request directly into TRACK, and those dedicated individuals will help you for fulfilling that. Another issue that came up originally was with environmental health and safety, trying to make sure that the research community was protected that was working with these samples. And so they've been developing guidelines in terms of how to do this. There have been some recent updates, and here under the tab New, Fresh, or Special Biospecimens, I put the URL for where you can see all of that information. Again, these slides will be posted, or someone can send this around in the chat, but there's guidance in this that's different than it was a little while ago. For those of you that have staff that aren't trained for the use of and handling of BSL-2 samples, there will be some additional training your staff members need to do, but this hopefully will help in terms of gaining more access to the samples that you want. The other thing we appreciated is that some of the samples that we have that are discarded specimens may not meet the sample requirements in terms of just the samples being as fresh, or the samples, just the integrity of the samples being what you'll need. And so I will encourage you, especially if you don't need a large number of samples, but very high quality samples, you may need to do prospective recruitment. And we have started doing inpatient recruitment so that we have the ability to get those prospective specimens for patients while they're in the hospital. Again, depending on what the clinical sort of circumstances you need, that may or may not be appropriate, but I encourage you, if you need to revise your application, to do that so we can get you the samples that you need. And then finally, separately, reviews for autopsy specimens are in process, and Dr. Goldman is leading those and is getting back to people in terms of that. There are several of the applications that did require—
You have 1 minute, Wendy.
Okay, multi-omic samples, and those are harder to fulfill. So as I said, if you can refine your applications, that's appreciated. Next slide. So just in terms of wrapping up, with this, as I said, there are samples that are available with priority to Columbia investigators. Make sure that you've got your staff prepared to be able to do this, and hopefully we've got ways of being able to get you what you need. I realize it's been slower, but hopefully those of you who needed letters of support for your grant applications, all of those should have been distributed. Okay, I think we've got time for questions.
So thank you, Wendy and Krzysztof. Do we have any questions from the attendees on the Biobank update? It looks like Stuart has a question.
Yeah.
Stuart, can you hear us?
Yes. Can you hear me now?
Yes.
Yep.
Yeah, I was just wondering if you could give any further information on the autopsy specimens and the availability on those. Seems to be a little bit of a bottleneck. What could we expect from those? We're interested in some of them, especially nasal autopsy
I
tissue.
think the best thing to do is directly contact Dr. Goldman, because he's the one handling the autopsy specimens.
Okay, great. Thanks.